This is going to be a quick lesson on the modification of RNA before it leaves the nucleus. So here's my cell, here's my nucleus, and this would be the cytoplasm. Cytoplasm contains a lot of enzymes, and so to protect the RNA before it leaves the nucleus, it gets modified a little bit. So here was my DNA, and through the process of transcription, one piece of RNA is made, just for one gene. So there is my RNA transcript. Before it leaves the nucleus, it gets modified. A couple things happen to it, 3 things in particular. The first thing that happens to it is it gets a 5' methyl cap. And that refers to a methyl always means it's got an extra carbon that has hydrogen on it. So it gets this hydrogenated cap on it, and it's a guanine. So if this, let me create a new screen. If this is my piece of RNA, the first thing that happens to it is this 5' cap. So I want you to think of it as like a little hat on the end of it. A cap is a hat. That protects the end from being degraded. Once it goes out into the cytoplasm, it could literally be digested by enzymes. That cap is a protection. The second thing that happens is the introns need to be removed. This process is called splicing, and it is done spliceosome. I'm not sure if that's spelled right or not, but a spliceosome is a complex that actually will remove sections of the RNA that don't count. So sometimes in our genes we have repeating sequences, and it'll go A, and it's like garbage. So the introns, I think of it as in the way. Introns actually stands for intervening. The introns have to be removed, and the exons, which get expressed, those are the important ones, those stay. So in a sequence, we could say that this is an exon, and this section here is an exon, and this section here is an exon. All right, so in my short little sequence here, I have 2 introns, 2 segments of RNA that actually don't code for anything. So this spliceosome is like this giant molecule that comes over the top of it, and it cuts it out. It removes the introns. So in the end, what's going to happen, this will be gone and this will be gone. My RNA is now shorter, still has the cap on, but now it's only this long. It has those intron sections removed. The third step is to add a poly-A tail. A is adenine, so at the end, they add on A to the end of it. So this will be the poly-A tail. A. That is backup to make sure that the production of the protein, once this goes to a ribosome, it's going to be used to build a protein. This says stop, enough. Don't code for any more amino acids. End the protein production here. So this is an extra, and you could also think of it as a protection. You got protection on this end, you got protection on this end, and it's a backup stop. Stop making the protein when you hit this point. So that's what a poly-A tail is for. So all of these 3 things are modifications that happen to the messenger RNA transcript before it leaves the nucleus. Once it is ready, so now this is ready, this can now leave. If you remember, a nucleus has a double membrane, but there are pores, there are little openings. It could go out through this pore and go to a ribosome. And when it goes to the ribosome, then we can have our final step, which is called translation. We can have translation begin at this point.